I'm going to present my computational work on putative coupling between dopamine receptor and calcium channels to investigate excitability properties in layer 2 stellate cells. So I'm working in neuroinformatics groups for Andrew Davison team at Paris Institute of Neuroscience. So we are doing the software development for Pi network, and also we are simulating a different microcircuit at cellular level for the brain. So this is a simulation work from our lab. So in introduction, you can see the right-hand side picture that we know that the grid cells are responsible for spatial calculation, and the MEC, the mid entorhinal cortex, is responsible for spatial cognition and memory. So the most of the cells in MEC is stellate cells, are in layer 2 is stellate cells. So they are useful in memory, cognition, and perception. Okay, so these stellate cells, and particularly the layer 2 stellate cells, these neurons are modulated by different subcellular signaling pathway like calcium dynamics, intercellular signaling pathways. And the most important thing is that, so we have the cell body, and after the soma, there is a segment that is known as initial axonal initial segment, AIS. So both soma and AIS in neurons are responsible for firing and modulating the action potential, so that can be transmitted to other neurons or other layer of the brain. Okay, so this AIS firing patterns. So basically, this region is rich with excitable ion channels, particularly sodium ion channels and calcium ion channels. So, and the density of these ion channels are high in this region, in AIS and the soma region. So, in different pathological conditions, maybe motor, Parkinson, Alzheimer, or perception cognition disorder. So now scientists are trying to find out the relationship of the AIS electrical activity with respect to those pathological conditions. So this topic is related to how the receptors, particularly the dopamine receptors, is modulating the electrical property of this AIS region. So we found that there are different type of dopamine receptors are there, D1, D2, D3, D5. So particular D2 receptors are present in this region, AIS region. And if you inject agonist, if you add agonist for D2 receptor, or because of dopamine, the excitability of this AIS region is changing. The excitability is related to particular ion channels, and then ion channels and the dopamine receptor pathway is modeled by our team. So we are going to show the modeling parameter, the modeling validation, and the predicted hypothesis for this pathway, AIS pathway. Okay, so we have choose particular one type of ion channel, that is T-type calcium channel, and D2 are receptors. Okay, so in this figure you can see the putative coupling between the D2R receptor. This is the D2R receptor. This is the AIS. This D2R receptor is modulating the T-type calcium channel conductance via virus pathway, CEM, PK, and AC pathway. So we are trying to model this pathway, and to nobody has done this modeling for the brain and for particularly this stellate cell. So we have added this model for the future application, and in future people can modify the model, add different parameters, and again find different hypotheses. Okay, so the method is like this. We have, as we have mentioned, this is single compartmental modeling. So we have different ion channels are there, sodium T-type calcium channel, L-type calcium channel, different potassium voltage gated potassium channel, then calcium activated potassium channels. And this is the electrical interpretation of this single compartmental cells. So we have converted the physical system to computational conceptual model, and then computational model. So we use neuron platform. That is the one of simulation tool of the eBrain community. Neuron also we have done. We are trying to do next as we are doing with Pine. We are simulating 3 or 4 platform software platform. But this is for neuron platform and single cell morphology. And all ion channels are modeled by either conventional HS formalism or Markov multiple state Markov mechanism. However, we are going to show this only one channel, that is calcium activated. Sorry, yes, the T-type calcium channel. Okay. So, what is the data we have taken from published literature and also from experimental work? Then we have developed equation for the diffusion-based T2 activation to alter bromo. Bromo is agonistic or D2 receptor, then PK and CMP concentration. So these three parameters are combined with differential equation to change the conductance of T-type calcium channel with respect to a particular dosage of this chemical composition. Then ODs are used for the mass action kinetics and biochemical reactions. So equations are derived assuming all reactions are in single compartment. We have not assumed that we are going to connect, the neurons are connected in a network. This is a single compartmental network. Okay. So result, as I have mentioned that we have done the one pathway lead to a receptor to T-type calcium channel. So here I am just showing 2, 3 pictures in result section. So first is T-type calcium channel internal kinetics. So here you can see the steady state activation and inactivation. The right hand side is the steady state activation, and this is inactivation curve of the T-type calcium channel. So when we are applying the PKI of 10 micromolar, this black, the red is the control. For the steady state activation, the this activation is shifting to right hand side. So right hand side means the T-type channel threshold is increasing, and the activation of T-type calcium channel is increasing with PKI of 10 micromolar. Okay. The same time, PKI is modulated by the CMP, and CMP is modulated by the D2 receptor. That means the D2 receptors, the higher dosage of D2 receptors can shift this steady state activation to positive side. But there is no change for the steady state inactivation curve. So it is similar both for the control and for the PKI. Okay. So next, after incorporating the ion channel, we have simulated the action potential for AIS region. All ion channels are there. Now we have simulated the action potential. You can see the control and the agonist of the D2 receptor, that is bromo. This is also 10 micromolar. The excitability of AIS region are simulated by a current clamp protocol. We have applied 400 picoampere current injection to evoke the action potential. So here also that in this figure we can see because of bromo or D2R activation, the shape is changing. You can see the red and here is excitability. The rising phase of the action potential is changing. Okay, because of the bromo. Okay. So next we have in addition to single action potential, we have simulated a series of action potential to investigate the frequency of the firing pattern with respect to control condition and with respect to this bromo admits D2 receptor agonist. So here you can see that the bromo, because of bromo, the number of action potentials are decreasing. That means for a particular current injection, the number of firing the number of spikes are decreasing because of D2R activation by modulating the T-type conductance. So the frequency is reduced after applying the CM, bromo, and PKI under the current clamp protocol. So in simulation, we have changed the dV by dt. In next figure, you can see because of the bromo, the dV by dt also changing and is decreasing. So this clearly shows that the signaling pathway is modulating the excitability property of the AIS region. That means the action potentials are the shape of action potential is changing, the frequency is decreasing, and it will cause pathological conditions. How to recover it? So we can use, okay, so this is the conclusion that because of the mechanism between D2 receptor and T-type calcium channels, the excitability is decreasing, and the PKI, CMP, and bromo are responsible for decreasing the conductance of T-type calcium channel. So as the T-type calcium channel conductance changing, the threshold to evoke the action potential is increasing. So that will cause pathological conditions, and to overcome this pathological condition, we have also, figures are not added, we have also tried that if you decrease or if you decrease the potassium channel conductance, or if you increase the L-type calcium channel conductance, then we can recover this dopamine effect, and that means we can use agonist of L-type calcium channel or antagonist of potassium calcium channel to overcome this problem. So in future, additional CMP formation and PK activation mechanism we are going to add for a more robust model, and all validations are done, and we have done parameter estimation from neuron protocols. So this is.
Thank you very much, Chitaranjan.